Good morning, students. In this video, I shall discuss the contribution of Morgan in the field of genetics. Morgan, the famous name in the field of genetics, his full name is T.H. Morgan, Thomas Hunt Morgan. He born in 1866 and died in 1945. And during this period, he contributed a lot to the field of genetics. Now, that is why for his contribution in the experimental genetics, he is known as the father of experimental genetics. Now, the famous experiment he conducted on Drosophila melanogaster, which is also called the queen of genetics, because this is the very good specimen or very good tools or material for the study of the genetics in the field of experimental genetics particularly. Now, why T.H. Morgan selected this material or selected Drosophila melanogaster as the suitable material for his experimental studies in the field of genetics? Now, it is also called the Cinderella, sometimes called it is the jackpot of genetics, whatever the case may be. But this material is very much suitable due to the following reasons. Then what are these reasons due to which he selected it? These are very easily available. These are very small miniature insects, normally cultured in a very small thermoplast. If you put some fruits here, or either it is banana or apple, when after within 2-3 days you will find the Drosophila here. So without any much effort, these are easily available everywhere. Now they are easy to culture. There is no any sophisticated culture medium required for the culture of this Drosophila. You just put some sugar material, then you will get the Drosophila easily there. Now, they have a very short life cycle. Within 15 days, they complete their life cycle. They lay their egg, and from the eggs, the new offsprings are produced. So that is why in a year you will get so many generations of the Drosophila, so that you can easily study their inheritance pattern of the different characters. Now, many eggs are produced at a time. Normally, the eggs which are large in number, that is why more number of offspring we can get, and from the offspring we can randomly select for mating, and that is why we can get the result very easily. And the large number of eggs also, it is very much easy to generalize the result. Now, it can be inactivated for study. Suppose we have to study the material or study the different characters under the microscope or under the high power magnifying glass or lens. So these specimens are normally Drosophila. They can be inactivated by giving certain anesthesia, and so it is easy to study there under the microscope. Now next one, the chromosomes, which are 4 pairs in number, and all the 4 pairs are clearly distinct from one another morphologically. So it is very much easy to distinct and easy to study the chromosomes under the microscope. Next, the sex chromosome, the XX, it is the female sex chromosome, and XY, it is the male sex chromosome, and the Y chromosome, it is distinct hook cell chromosome. So that is why it is very much easy to distinguish the male organism from the female organism from the chromosome itself. So that is why it is another important character for its selection. Now breeding occurs throughout the year. Suppose the life cycle is very short, no doubt within 15 days. So in a year, without any season, so some organisms are seasonal in nature. That means they only reproduce in a particular season. But this Drosophila, it breeds throughout the year. So that is why we can get the result, get the offspring throughout the year, and we can study once, not once, twice or thrice, but many times in a year. Now, the polytene chromosome, a special type of large-shaped chromosome present in the salivary gland of the larva of this Drosophila, so that These large polyethylene chromosomes are very much easy to study or detect their defect or abnormality in the chromosome. So that is why this Drosophila, it is one of the suitable material which were actually for the first time used by Morgan for the study of the transmission patterns of the different characters. So that is why Drosophila is called the queen or the Cinderella or the jackpot of genetics. T.H. Morgan, the famous scientist who conducted his experiment on Drosophila melanogaster, and his main contribution in the field of genetics, that is the linkage. Linkage is one of the important phenomenon which was not earlier mentioned by Mendel. According to Mendel, all the genes are independently associated from one generation to the next generation. But for the first time, Morgan explained that some genes are not separated independently, but they are transmitted from one generation to the another generation simultaneously together. So that is why this phenomenon is called linkage. So the genes which are present in the same chromosome, they normally do not separate from one another, and they pass from one generation to the next generation combinedly or with one another. So that is called linkage phenomenon. Then are these genes actually transmit in the same linkage manner? No. Sometimes they are also separated from one gene to another, or one gene and another gene, they are separated from one another and they transmitted separately to the next generation. This phenomenon occurs normally during the sexual reproduction, and that phenomenon is called crossing over. We shall discuss it later on, and this crossing over phenomenon was also discovered by Morgan. Now, what are the linkage maps? How the different genes which are present in the same chromosome, they are present in equal distance in a linear manner. So this is called the linkage map, or it is called chromosomal mapping. And he done it, and according to him, that is the 1 millimorgan, centimorgan. These are the units now given, and this phenomenon was also discovered by Morgan. That is why he received the Nobel Prize in 1933. And after that, there are so many other phenomena like variations due to sexual reproduction, or during sexual reproduction when the gametes are formed, how the different genes are actually isolated from one another, and they are going to the different gametes, and these gametes again fuse together to form the new combination. And this type of variations which are occurred from the parents to the offspring. Suppose the parents, they have certain character, but in the offspring, some types of variations is observed, and these variations are due to sexual reproduction. And during sexual reproduction, what happens? The meiosis occurs, and during meiosis, what happens? Now crossing over occurs between the linked genes. The crossing over phenomenon occurs, and due to this crossing over, some sort of variation occurs in the offsprings. That is why the offsprings are not exactly similar to their parents during sexual reproduction. In case of asexual reproduction, normally the offsprings are genetically similar, but in case of sexual reproduction, the variations are due to this crossing over during the meiosis when the gamete formation occurs. Now, he also discovered this linkage and distinguished the linked genes and the unlinked genes. The genes which are present in the same chromosome are the linked genes, and the genes which are not in the same chromosome are called unlinked genes. But the linked genes are also sometimes separated from one another by the process of crossing over. Now, he also given the chromosome theory of linkage. Now, what is this chromosome theory of linkage? This chromosome theory of linkage means what? All the genes which are responsible for the expression of the characters in the offspring, these genes are present or located in the chromosome. Suppose this is one chromosome, and in this chromosome, the genes are located in a linear manner or arranged in a linear order. And these genes, each gene is responsible for the expression of one character in the offspring. So this was called the chromosome theory of linkage, and this phenomenon was for the first time explained by T.H. Morgan. Now another important thing, that is the gene theory of inheritance. Now what is this gene theory of inheritance? Now specific genes are associated in the specific chromosome. So that is why the genes which is responsible for the expression of at least one character, but these genes are always present in a specific chromosome. Suppose a chromosome number 1, it contains these characters, that means the genes for this particular character is present there in particular chromosome. So that is called your gene theory of inheritance. So according to this, the genes are always located in the same chromosome, generation after generation. So that is why they express the character, and if there will be any defect or any change in that particular chromosome, that character is affected in the offspring. Now, next, the frequency of the crossing over between the two linked genes. Suppose this is a chromosome and these are the different genes present there in the chromosome. Now, when and how the genes are actually separated from one another during crossing over. During crossing over, no doubt, these two genes, they are separated sometime. Now, how and when they are separated? The frequency of crossing over. Suppose the crossing over occurs between these non-sister chromatids. So, what is the chance of this crossing over between the two genes? Suppose this is one gene is here, another gene is here. One gene is here, another gene is here. Then what is the chance of separation of, suppose gene 1, gene 2, and here gene 3 and gene 4. Gene 1 and gene 2, they are wide apart from one another. So that is why the chance of crossing over or the frequency of crossing over between two linked genes is directly proportional to the distance between the two genes. Suppose this is gene number 1 and this is gene number 2, and the distance between these two genes is very large. So that is why the frequency or the chance of crossing over is more in comparison to the 3 and 4, because the 3 and 4, they are very close to one another. So that is why the chance of crossing over in this particular point is very low, because the distance is short. So that is why chance of crossing over is less. So that is why they normally do not separate from one another, and they normally pass simultaneously or together from one generation to the another generation. Now this type of processes which are going on, they are during the transmission of character or transmission of gene from one generation to another generation. That is the linkage and the crossing phenomenon, and it was the famous discovery of T.H. Morgan. Nowadays we have already cracked so many other things, but the founder or the pioneer worker on the experimental genetics was no other than T.H. Morgan, and we should always remember him for his contribution towards this field of genetics. Now, I hope this video is very much useful for you, and if it is really useful for you, then please like it and subscribe for further videos. Thank you.